Chapter 21. Reconstructing Membrane Integrity. Membrane integrity is not restored by a single input or isolated intervention. It is rebuilt through repeated cycles of lipid incorporation, structural organization, and selective removal of damaged components. The membrane exists as a continuously updated interface where composition determines behavior. Reconstruction begins when the input pool, oxidative environment, and energy availability align to support stable assembly. From that point forward, each remodeling cycle shifts the structure incrementally toward a configuration that maintains containment, supports signaling precision, and resists degradation under load. The bilayer must reestablish uniform packing. Phospholipids arrange according to the properties of their fatty acid chains, and the distribution of these chains determines how tightly the membrane holds together. When stable lipids dominate, adjacent molecules align with minimal gaps, reducing passive permeability and allowing gradients to persist. This affects not only ion balance, but also the mechanical behavior of the membrane, providing a consistent platform for embedded proteins. Reconstruction, therefore, involves replacing irregular arrangements with a more uniform lattice that supports controlled exchange. Domain organization returns as composition stabilizes. Membrane microdomains form when specific lipids and proteins cluster into regions with distinct properties. These regions serve as sites for signaling, transport, and structural coordination. When lipid composition is inconsistent, these domains become unstable or fail to form altogether. As stable lipids are incorporated, the membrane regains the ability to segregate into organized regions, allowing receptors and associated proteins to localize correctly. This restores the conditions required for coordinated signal initiation and propagation. Cholesterol distribution becomes more effective within this reconstructed environment. It positions itself between phospholipids to regulate spacing and maintain consistent fluidity. In a stable membrane, cholesterol prevents excessive movement without restricting necessary interactions. This contributes to both mechanical stability and signal fidelity, as receptors operate within an environment that supports predictable behavior. Membrane proteins begin to function within defined constraints. Transporters, channels, and receptors depend on the surrounding lipid environment for their structure and activity. When that environment is restored, these proteins regain their ability to operate efficiently. Ion channels maintain gradients with less energy expenditure, receptors respond proportionally to stimuli, and transport systems move substrates in a controlled manner. The improvement is not due to changes in the proteins themselves, but to the restoration of the conditions that allow them to function as designed. Mitochondrial membranes follow the same principles. Reconstruction of the inner membrane improves the containment of proton gradients and stabilizes the arrangement of electron transport complexes. As leakage decreases, ADP production becomes more efficient, reducing the need for compensatory increases in substrate oxidation. This restores a more stable energy supply, which supports maintenance and repair processes throughout the cell. Repair mechanisms operate more effectively as structural integrity improves. Damaged lipids are identified and replaced, and oxidative chain reactions are limited by a membrane that resists propagation. The rate of damage decreases, allowing repair processes to outpace degradation. Over time, this shifts the balance toward maintenance rather than continuous correction. Intercellular consistency emerges as membranes across tissues converge towards similar structural properties. This reduces variability in signaling and transport, allowing coordinated responses to systemic inputs. Hormonal signals produce more uniform effects, and energy distribution becomes more predictable. The system begins to operate as an integrated whole rather than as a collection of independently behaving compartments. Reconstruction proceeds through time and repetition. Each cycle of lipid turnover contributes to the overall structure, and the pace of change depends on the rate at which membranes are replaced. Sustained conditions are required to ensure that new structures are not immediately disrupted by unfavorable inputs. As stable composition becomes the default, maintenance requires less intervention, and the system holds its structure with greater efficiency. Reconstructing membrane integrity, therefore, involves restoring the physical conditions that define how cells interact with their environment. By aligning composition, reducing damage, and supporting energy production, the membrane regains its role as a controlled interface. From this foundation, transport, signaling, and metabolic processes realign, producing outcomes that reflect the underlying architecture rather than compensatory adaptations to instability. Reintroducing stable lipid substrates. Membrane reconstruction depends on what is available for incorporation at the moment of remodeling. Once unstable inputs are reduced, the system requires a consistent supply of lipids that support tight packing, controlled fluidity, and resistance to degradation. These substrates do not act as signals in isolation. They become part of the membrane itself, altering its physical properties with each cycle of replacement. Reintroduction therefore defines the direction of rebuilding by determining which structures are repeatedly selected and embedded into the bilayer. Saturated fatty acids provide the foundation for this process. Their linear structure allows adjacent lipids to align closely, reducing spacing and limiting passive permeability. As these lipids are incorporated into phospholipids, the membrane becomes more capable of maintaining ion gradients and resisting deformation under stress. This improves the baseline conditions under which transporters and receptors operate, as the surrounding environment becomes more uniform and less susceptible to fluctuation. Monounsaturated fatty acids introduce controlled flexibility into this structure. A single double bond creates a predictable bend that prevents excessive rigidity while preserving overall packing integrity. This allows membranes to maintain mobility where it is required for receptor interaction and domain formation without losing containment. The balance between saturated and monounsaturated lipids defines a range of fluidity where both stability and function are preserved. Cholesterol integrates with these lipids to regulate spacing and mechanical behavior. It inserts between phospholipids and adjusts local packing based on surrounding composition, preventing extremes in fluidity. As stable lipid substrates become more prevalent, cholesterol can perform this regulatory role more effectively, contributing to consistent membrane properties across different tissues and conditions. Phospholipid synthesis reflects the availability of these substrates. Enzymatic pathways assemble phospholipids using fatty acids drawn from the circulating pool. When stable substrates dominate, newly formed phospholipids inherit properties that support membrane integrity. Over successive remodeling cycles, this shifts the overall composition toward structures that maintain gradients and support organized signaling. Incorporation occurs gradually. Existing membranes are not replaced immediately, but are remodeled through continuous turnover. Each cycle introduces a portion of new lipids while removing older or damaged components. The rate of change depends on tissue-specific turnover, meaning that some systems reflect the new substrate profile sooner than others. Sustained availability of stable lipids ensures that each cycle contributes to a consistent direction of change. Signal behavior begins to align with these structural changes. Receptors operate within environments that allow proper clustering and conformational shifts, improving the translation of external signals into intracellular responses. Transport systems maintain gradients with reduced energy expenditure, and mitochondrial membranes contain proton gradients more effectively, increasing ADP production efficiency. Resistance to oxidative damage increases as stable substrates replace more susceptible lipids. With fewer sites for reactive species to initiate chain reactions, the rate of membrane degradation decreases. This allows repair mechanisms to maintain integrity without being overwhelmed, reinforcing the stability established through composition. Distribution of these substrates across tissues is mediated by lipoprotein transport systems. As circulating lipid composition changes, delivery to different organs reflects this new profile. Over time, this leads to a convergence of membrane properties across tissues, reducing variability in function and improving coordination between systems. Reintroducing stable lipid substrates therefore provides a material basis for reconstruction. By supplying lipids that support consistent packing and controlled flexibility, the system rebuilds membranes that maintain containment, enable precise signaling, and sustain efficient energy production. The outcome emerges from repeated incorporation rather than immediate effect, as each cycle of remodeling integrates these substrates into the architecture that governs cellular behavior. Saturated and monounsaturated rebuilding. Reconstruction of membrane integrity depends on reestablishing a lipid environment that can maintain both containment and controlled interaction. Saturated and monounsaturated fatty acids form the structural base that allows this to occur. Their integration into phospholipids determines how tightly the bilayer holds together, how much lateral movement is permitted, and how consistently proteins embedded within the membrane can operate. The rebuilding process is not about increasing one class in isolation. It is about restoring a ratio that produces predictable mechanical behavior across the membrane surface. Saturated fatty acids provide the primary framework. Their straight hydrocarbon chains align closely with adjacent lipids, reducing gaps within the bilayer and lowering baseline permeability. As these lipids accumulate through remodeling cycles, the membrane becomes more resistant to uncontrolled ion and proton movement. This strengthens gradient maintenance in both cellular and mitochondrial membranes, directly affecting transport efficiency and energy capture. The increased packing also stabilizes the physical platform on which receptors and enzymes operate, reducing variability in how they respond to input. Monounsaturated fatty acids introduce a controlled degree of flexibility into this framework. The presence of a single double bond creates a consistent bend that prevents excessive rigidity without disrupting overall organization. This allows membranes to retain the ability to accommodate protein conformational changes and lateral movement required for signaling. When incorporated alongside saturated lipids, monounsaturated fatty acids define a functional range of fluidity where receptors can cluster, separate, and reassemble without losing structural coherence. The interaction between these two classes determines membrane behavior under varying conditions. Saturated lipids anchor the structure, while monounsaturated lipids modulate spacing to maintain functional mobility. When this balance is achieved, membranes resist deformation under mechanical stress and maintain consistent permeability across temperature changes. This stability allows cellular processes to operate within defined constraints rather than adapting continuously to fluctuating structural conditions. Cholesterol integrates into this rebuilt environment as a regulator rather than a compensator. Within a membrane composed of stable substrates, cholesterol fine-tunes local packing by preventing excessive fluidity and limiting rigid crystallization. This maintains uniformity across regions of the membrane, supporting the formation of organized domains where signaling proteins can localize and interact. Without a stable base of saturated and monounsaturated lipids, cholesterol cannot perform this role effectively, and its distribution becomes uneven. Mitochondrial membranes reflect the same principles. As saturated and monounsaturated lipids replace more permeable structures, the inner membrane regains its ability to confine the proton gradient generated by electron transport. Reduced leakage increases the proportion of energy captured as ADP, lowering the need for compensatory increases in substrate oxidation. This stabilizes cellular energy supply and supports processes that depend on consistent ADP availability. Receptor function improves as the surrounding lipid environment becomes more consistent. Insulin receptors, ion channels, and other membrane proteins rely on a stable platform to undergo conformational changes and interact with downstream components. When the bilayer provides uniform mechanical resistance and controlled fluidity, these proteins operate with greater precision. Signal initiation, amplification, and termination align more closely with input, reducing the variability that characterizes unstable membranes. Remodeling cycles gradually embed this balance across tissues. Each cycle replaces a portion of existing lipids with those available in circulation. Sustained availability of saturated and monounsaturated substrates ensures that new membrane segments reflect the desired structural properties. Over time, variability between tissues decreases as composition converges toward a common baseline that supports coordinated function. Resistance to oxidative damage increases as these lipids become predominant. With fewer highly reactive double bonds, the likelihood of chain reactions decreases, allowing membranes to maintain integrity under metabolic and environmental stress. Repair systems operate more effectively because the rate of damage is reduced, reinforcing the stability achieved through composition. Saturated and monounsaturated rebuilding, therefore, establishes the mechanical conditions required for consistent cellular behavior. By providing a foundation that supports tight packing and controlled flexibility, these lipids allow membranes to maintain gradients, organize signaling domains, and resist degradation. The system regains coherence as each remodeling cycle integrates these properties, producing a structure that supports efficient and coordinated function across tissues. Time-dependent membrane replacement. Membrane reconstruction unfolds according to turnover rates that differ across tissues, not according to the timing of any single input. Each membrane is continuously renewed through cycles of degradation and synthesis, where existing phospholipids are broken down and replaced with new ones assembled from circulating substrates. This process operates at a pace defined by cellular activity, metabolic demand, and exposure to stress. When the substrate pool shifts towards stable lipids, the effect is not immediate because existing structures remain in place until they are replaced. Change appears as a gradual transition where newly incorporated lipids alter local properties first, then spread across the membrane as turnover continues. The replacement process begins at the level of individual phospholipids. Enzymatic pathways remove fatty acids from existing molecules and re-esterify them with those available in circulation. This selective exchange allows the membrane to adjust composition without requiring complete dismantling. As stable substrates become more available, these enzymes incorporate them into new phospholipids, progressively altering the characteristics of the bilayer. The rate of this exchange determines how quickly structural changes manifest within a given tissue. Turnover varies significantly between cell types. Tissues with high metabolic activity, such as the liver and intestinal epithelium, replace membrane components rapidly due to constant exposure to substrates and environmental inputs. These tissues respond more quickly to changes in lipid availability, showing early shifts in permeability, signaling behavior, and energy handling. In contrast, tissues with slower turnover, such as neural structures and long-lived cells, require extended periods of consistent input to achieve comparable changes. Their membranes retain previous composition longer, delaying full reconstruction. Mitochondrial membranes follow a similar pattern but are influenced by both lipid turnover and organelle dynamics. Fusion and fission processes redistribute membrane components across the mitochondrial network, allowing localized improvements to spread over time. As new lipids are incorporated into individual mitochondria, these dynamics facilitate the gradual normalization of membrane properties across the entire population. The cumulative nature of replacement defines the trajectory of stabilization. Early changes may be subtle, affecting only small regions of the membrane or specific cellular functions. As successive cycles incorporate stable lipids, these regions expand and the overall behavior of the membrane shifts. Gradients become easier to maintain, receptor function becomes more consistent, and energy production stabilizes. These improvements do not occur in isolation but build upon one another as the proportion of stable structure increases. External conditions influence the pace of this process. Continued exposure to unstable or oxidized lipids slows progress by reintroducing components that counteract reconstruction. Conversely, maintaining a consistent supply of stable substrates allows each cycle to contribute to the same direction of change, accelerating convergence toward a stable configuration. Energy availability also affects turnover. Membrane synthesis, repair, and remodeling require ATP. When mitochondrial efficiency improves, these processes can proceed at a rate that supports effective replacement. When energy is limited, turnover slows, prolonging the presence of unstable structures. Time-dependent replacement therefore governs how structural changes propagate through the system. The body does not rebuild membranes instantly, but through continuous, incremental updates that reflect the current substrate environment. As stable lipids are incorporated across successive cycles, the membrane transitions from a variable and permeable state to one that maintains consistent packing, controlled exchange, and reliable signaling behavior across tissues. Restoring electrical and signaling stability. Electrical behavior in biological systems is defined by how well membranes separate charge and control ion movement. Every gradient across a membrane represents stored potential that can be used to drive transport, generate signals, or sustain cellular work. When membrane structure becomes inconsistent, this separation weakens. Ions cross more freely, gradients dissipate, and the baseline electrical state of the cell becomes unstable. Restoring stability requires rebuilding membranes that can maintain charge separation with minimal leakage, allowing electrical properties to remain consistent across time and across tissues. Ion channels and transporters depend on this environment to function correctly. These proteins do not generate gradients on their own. They regulate the movement of ions according to the conditions established by the membrane. When permeability increases, passive ion flow competes with controlled transport, forcing channels and pumps to work continuously to maintain balance. This increases energy demand and reduces the precision of electrical signaling. As membrane integrity improves, passive leakage decreases, and transport systems regain the ability to establish and maintain gradients with less effort, stabilizing the electrical baseline of the cell. Membrane potential becomes more consistent as structural integrity is restored. The difference in charge between the inside and outside of the cell depends on selective permeability and active transport. When the membrane supports tight packing and controlled ion movement, this potential remains stable, providing a reliable platform for signaling events. Fluctuations decrease, and the cell can respond to inputs without interference from background instability. Receptor function is directly influenced by these electrical conditions. Many signaling processes depend on changes in membrane potential or on the local ionic environment. When the baseline is unstable, receptor activation becomes less predictable, and downstream signaling pathways receive inconsistent input. Stabilizing the membrane restores the conditions required for receptors to respond proportionally to stimuli, improving the clarity of signal transmission. Synaptic and neuronal signaling rely heavily on these principles. Transmission of electrical impulses depends on rapid, controlled changes in membrane potential. When membranes cannot maintain consistent gradients, signal propagation becomes irregular. This affects timing, amplitude, and coordination of neural activity. As membrane properties normalize, conduction becomes more reliable and communication between cells regains precision. Mitochondrial membranes contribute to electrical stability through their role in maintaining proton gradients. These gradients represent a form of electrical potential that drives ADP synthesis. When leakage is reduced, the gradient remains stable, supporting consistent energy production. This ensures that energy-dependent signaling processes have a reliable supply of ADP, further stabilizing cellular behavior. Signal termination improves as stability returns. In a stable membrane, channels close, receptors disengage, and gradients are restored to baseline conditions in a controlled manner. When structure is inconsistent, these processes are delayed or incomplete, leading to prolonged or overlapping signals. Restoring membrane integrity allows signals to begin and end within defined time frames, reducing interference between pathways. Coordination across tissues depends on the consistency of these electrical properties. When membranes across different cell types maintain similar baseline conditions, signals transmitted through neural and endocrine systems produce aligned responses. Variability decreases, and the system operates with greater coherence. Restoring electrical and signaling stability, therefore, depends on rebuilding membranes that can maintain charge separation, regulate ion movement, and support predictable receptor behavior. As leakage is reduced and gradients are preserved, the system regains the ability to process and transmit signals with precision, allowing coordinated function to emerge from a stable structural foundation.